Hi, this is Miss Slinton, and this is Honors Bio, 7th period review for Unit 2 exam. Say hi. Hi. Very good. All right, now we're going to do two things here. Number one, I need to review chapter 27, but then I have a kind of a whole review together for chapters 1 through 4. Okay, so we have two parts to it. On chapter 27, this was the early Earth. I would be comfortable with how old the Earth is. How old is the Earth?
14 billion.
No. How old is the Earth?
4.6.
Yes, 4.6 what?
Billion
years. Good. And then I would be familiar with how, oh, there we go. And then how old is the first cell?
3.6.
So it goes 4 and then it goes 3 billion, right? So keep that, thank you, sweetheart. Keep that in mind. And we're going to go through that, but I just want to make sure you have those two dates. We talked about early Earth and about plates moving, right? We talked about what moves those plates. Convection currents. So you want to review that. You also want to review the different types of dating, and one of them we talked about absolute dating and in relationship to providing some energy. Then we talked about absolute dating. We talked about isotopes giving energy as well as gravity to movement of the hot iron and giving movement to those convection currents. But then we talked about absolute dating on how fast an isotope breaks down is referred to as its what? Half-life. Okay, so we know right now how much, when we look at carbon-12, when we look on the periodic table, it doesn't just say 12, it says 12 point what? 011. Oh, you don't know that, that's okay. But, no, I'm sorry, I'm expecting you to know that. So when you look at that's because it's giving you all the isotopes that are there. What is an isotope, by the way? When you vary the number of neutrons. Yes, you left your computer on and it was put away for you and logged out, so you need to not ever leave the classroom without making sure you log out. Okay? Now, back to me. So when you look at a sample, you can find out how old it is by looking at the percentage of the isotope in the sample versus the percentage of the isotope currently as it's found. Okay, and that is absolute dating. And then the next thing, going quickly because there's so much, we went through the different kinds of plate boundaries. So you want to make sure that you're comfortable with those. We talked about divergent boundaries. Where do you find divergent boundaries all the time?
When a continental plate meets a oceanic.
No, that would be convergent boundary. Where do we find divergent boundaries? Seafloor spreading. He's not even in my class. I love you. Seafloor spreading. And so then you're going to have a divergent boundary, right? Convergent boundary is what you were just talking about, where maybe an oceanic plate meets a continental plate, and the oceanic plate does subduction underneath the continental plate. Then it melts, and then you could get a what on the continental plate?
Volcano.
Volcano, good. And then transform plate is like where we're sitting right here. Pacific and North American plate going past one another, causing a little bit of wrinkling here, which could lead to what? Earthquakes. Yep. Okay, so you want to make sure you review those. We had evidence as well for the different types of the plate tectonic theory to support that. We looked at the reversal of the iron in the seafloor spreading and the magnetism, and we looked at depression underneath glaciers or bodies of water, and we looked at how the continents all fit together like a puzzle piece. And you could see fossils, a continuous fossils from one plate to another plate, which would then kind of, if you put them together like in the puzzle pieces, you can see the continuity of that. Then the way we ended, I need you to not talk when I'm talking, the way we ended that was we talked about how when life is evolving on Earth, it can change the surface of Earth, the geosphere of Earth. When the geosphere changes, it can have an influence on what life forms you have and vice versa. So you have that continual feedback one on the other. Okay, so that was the setup that told us too that early Earth was when we look at how life evolved. And again, there is a whole review on this already. I'm just going through this very rapidly to remind you of it if you haven't started looking at that, but we have reviews on this already. When we look at the story of life, the gases that you would find on the early Earth are similar to gases that would come out of a volcano. And what gas was missing? Oxygen, no free oxygen, which was actually very, very helpful because when you have oxygen in the air, it oxidizes things, it breaks things down. So by having a reducing environment, no free oxygen, this is conducive to building. And then from there, we went through the steps of life. Okay, if you remember this part, we talked about the inorganic molecules in the outgassing, and then there would be rain. This would be, these similar gases would be in the early oceans. And we wanted to build organic molecules. What would be some energy we would have available for us to build those early organic molecules? What? Lightning and UV radiation, molten magma. And from those energy forms, you could build our first organic molecules. The building blocks, which we learned about later, right? We did quite a bit of study on those. What were the 4 bricks we talked about? Okay, so nucleotides, right? Base sugar phosphate, nucleotides, what else? Amino acids. Fatty acids. We talked about glycerol. What else? One more. S. Steroids are a type of lipid. One more. Sugars. Yeah. So from there, you could build these organic molecules, and then our idea would be to take these organic molecules, these monomers, and have them become what? Polymers. But currently, we use enzymes to do that. But an enzyme is a polymer, so we don't have those available. Again, what kind of energy could be provided for that? Lightning, radiation, molten magma, yes. Clay can act as a catalyst and also to facilitate those reactions. Once we have our polymers, then we can start to build our cells. Was our first cell a prokaryotic or a eukaryotic cell?
Prokaryotic.
You think it was an autotroph or a heterotroph? Heterotroph, because it's easier to consume nutrients. So you just want to kind of understand the sequencing of it and maybe evidence to support that. Okay? One piece of evidence to support that was a man named Miller did an experiment, and just within less than 2 weeks, he took the gases that might have been on early Earth. He had this circular apparatus where the electrode would act like what? Lightning. And then he was able to make these organic molecules. Okay, and remember, how much time do we have from the beginning of Earth until we had our first life form?
A billion.
A billion years. And he was able to get from step 1 to step 2 within just 2 weeks. So we had, basically a billion years to do the rest of the steps. Okay, so that would be some evidence for that. Also, we know that when hot proteins cool or lipids are in water, that you can get these sphere shapes. And we now know from chapter 4 that what is a membrane built out of? Lipids and proteins, yeah. And so here, these are operans, quasarbate droplets. This is Fox's proteinoid microspheres. These are like little protein spheres that are the beginning of a barrier. And I asked you a question on Google Classroom. Did you see that question I asked?
Yeah, I answered.
What do you think is what makes membranes, and this goes all the way back to this chapter in 27, why are they so important, do you think?
Because it keeps it.
Yes.
Because it protects the cell and it keeps it all together.
Protects the cell, keeps it all together. I'm looking for one more thing. It creates a what? A boundary. What's another word for that? Barrier. So what's going on inside is different than what's going on outside. Because that membrane, that phospholipid bilayer, you can't cross it unless you are larger charged. And if you're larger charged, you have to use a channel or a carrier or endocytosis. So you're controlling what's going on here, which allows that more likely to have isolated reactions that could lead towards the evolution of life. Okay, so we have that barrier. So just review those steps. There's some pictures in your book as well, and then back to here again. Okay, so that is chapter 27. So it's a little bit out of sequence, but I'm using that so that when we move in to talk about the characteristics of life, here are the top 10 things you want to know about. Notice I put plus early Earth and life. That's chapter 27. This right here are chapters 1 through 4, right there. Yeah, I knew you'd want that. What?
Just wanted a little bit.
A little bit? So let's hit the highlights on this. What do we know? What are we struggling with? Characteristics of life. Let's do that really quickly. Ready? Here we go. Living things are organized, made of cells. Stop. Do it faster. Living things are organized, made of cells. They acquire materials and energy. With me, they maintain homeostasis. They respond to stimuli and reproduce. They grow and develop, and they adapt and die. And when I say living things are organized, how are they organized? Atoms, molecules, organelles, cell, because this is where life starts, tissue, organ, organ system, organism, population, community, ecosystem, biosphere. Now, when we say organism, we can take all organisms and put them into 3 bins. What are the 3 bins? We can take all organisms and we can put them into it. Bacteria, archaea, eukarya. Bacteria, archaea, and eukarya. Eukarya are anything that's built out of a eukaryotic cell, right? And there are 4 kingdoms that are built out of eukaryotic cells. What are they? Animalia, Protista, fungi, and plantae, right? Bacteria and archaea, they have something in common. They are both what? Prokaryotic. What's different about them? Bacteria are found everywhere, and archaea are extremophiles and more closely related to eukaryotes. And if I say that, bacteria, archaea, eukarya, what classification category is that?
Kingdom. Domain.
No, domain. And it goes domain, kingdom, phylum, class, order, family, genus, species. Did King Philip come over for good soup? Remember that? And we learned man's classification.
Do we have to?
What do you think?
Yeah.
Okay. Okay, so look what we did right here. Check. We just did characteristics, organization, and classification of life. Did we not just go through that? What domain is man in? What domain are we in? Eukarya. Domain, kingdom. What kingdom are we in? Animal kingdom. What phylum? Chorodotic. Did King Philip class? What class are we in? Look at all the mammals. Order.
Bad monkey.
No monkey. And we remember primates. Hi. See, she knew right away. All I had to do was look at her. We knew.
I actually just wanted to come in and give you a hug. So I have to step out now because I think I have to go somewhere else.
Okay. I miss you. You're super smart. Okay.
You're all super smart.
They are. Okay, domain, kingdom, phylum, class, order, no monkey, primate, right? Family, Hominidae, genus, species, what is it? Homo sapien. Perfect. Okay, so that's classification of life. Check, done, boom. Okay. Okay. There it goes. All right, next, atomic structure. The atom story. Atoms are made of protons, neutrons, and electrons. The protons and the neutrons are in the nucleus, atomic mass. The electrons are in energy levels. 2, 8, 1832. 8, 8. 8 is great. Octet rule. Perfect. Okay. So we know atomic structure. Now, we know if I look at an atom, its behavior is totally dependent on the number of valence electrons. Because they're always trying to get happy. What are the 2 ways to get happy? Fill it up, right? Fill it up or get it to 8. Now, in order to do that, they might enter into some bonds. What are the bonds that we learned about? Covalent bond, when you share to get happy, and ionic, where I go, I'm not happy with these electrons, I'll get rid of them. You catch them, you're happy, I'm happy, now we're happier together, right? Okay, but then we also learned about other bonds too. What's another bond? Hydrogen bond. Where's a good place to see hydrogen bonds?
Water.
Water. Because water has polar covalent bonds between the oxygen and the hydrogen. right? Oxygen really, it shares with hydrogen, but not very well. So it has a strong pull on the electrons and pulls it towards it. That makes hydrogen partially positive, oxygen partially negative. This sets up this partial charges, so when water molecules are all together, opposites attract. The partials attract. Yes?
So the partials, is that the reason for surface tension?
Yes, because the partials set up hydrogen bonds, and that's the reason for surface tension. Then we talked about the properties of water. High heat capacity, right? Can hold a lot of heat. High heat of vaporization, right? We talked about cohesive, adhesive. We talked about ice is less dense, the solid when it freezes. You want to review those properties of water because they're important. Okay? Okay, we did 3. I didn't do, what's an ion when you change what?
Number of electrons.
Electrons, and isotope when you change the number of neutrons. This one will affect the atomic mass, but this one won't, right? Okay, so we did that. Oh, pH is a measure of the number of what? Hydrogen. You're not going to remember that?
The hydrogen.
Okay, the number of hydrogens. So when your hydrogen number is high, you have a low pH, and it's acidic. Okay? All right, organic molecules. The polymers and the monomers that build them. So first of all, I would know, what are the 4 polymers we talked about? What's the first polymer we talked about? No. Carbohydrates, and they're built out of sugars. Okay? Then the second one we looked at was lipids. What were the 2 categories of lipids? Fats and steroids. Steroids are the 4 rings. Where's somewhere where we have seen steroids? Cholesterol in a cell membrane. What kind of cells? Animal cells. Good. And then fats, we said there were 2 kinds. What were the 2 kinds? Saturated and unsaturated. Trans fat is when you take an unsaturated, supersaturated with hydrogen to make it a solid. Okay. Which reminds me, do your additional objectives. They will be on the test. Okay, so make sure you review them. Okay, make sure you know, yes. What?
Even chapter 4? That wasn't
All of them on there, yeah. And then I just didn't ask you about it in the right spot. I gave it to the wrong chapter is what I did. Okay. Next, okay, we've done carbohydrates, we've done proteins, we've done lipids. Let's do proteins. Proteins are built out of amino acids. I would know how to draw an amino acid. Do you know how to draw one?
No.
Because I told you needed to. Remember we did little simple diagrams of them, I believe?
Oh yeah, with the
Okay.
carbons being.
So if I was going to do a simple sugar, it would look like this, right? A sugar. And you can put these together to build carbs, right? And amino acids have a central carbon, H, remember R group, NH, H, C double bond O, OH. Remember that? We hook those together to build to build proteins. And this is an amino acid. How many different R groups do we have?
20.
20. Okay, there you go.
So that's the amino acid, is the R.
No. What's the amino acid? This whole thing is an amino acid.
Yeah, that's what
Not the R group. That's just a part of it. So
just
then you hook amino acids together to build protein. Then what else did we learn? We talked about glycerol. Now glycerol is a 3-carbon. I don't know that I would have you draw that. And then it has long, what are these?
Fatty acid.
Fatty acid chains. And those fatty acid chains could either be what? Saturated or unsaturated. Where do we find saturated ones? Animals.
Straight.
And unsaturated is from plants.
Plants. Which
It's like
one's a
liquid.
solid?
Saturated.
Yeah, good. Okay, then we talked about nucleotides. Nucleotides consist of 3 things, right? Oh, and here's cholesterol. This would be like simplified diagram for a steroid. Okay, then we talked about nucleotides. What do nucleotides consist of? 5 carbon sugar.
Phosphate group.
Yeah. Phosphate. What else?
Nitrogen base.
Nitrogenous base.
Same thing. Yeah.
Wasn't really arguing. Okay, so those 3 things together then will form a nucleotide. Nucleotides form DNA, RNA, and ATP. If any of this is scary to you, go back and watch the reviews just on the 4 important organic molecules, right? Okay, so then the other thing I would want to review is how did we hook those monomers together to build polymers? What was that process by which you could take one sugar and hook it together with another sugar? How did you do that?
Dehydration.
Yes, dehydration synthesis. Dehydration synthesis would build. And as a result, when we dehydrate them, we're taking away what? Water, so we can hook them together. Right? So because we have to create a need, they're fine by themselves. Take away an H and an OH, one H from one and an OH from another, and then we can bring them together. And the opposite of that is called hydrolysis. You have to add in water to break them up. If you're going to pull them apart, you got to give them something to pull them apart. So water's going to break those up. That's hydrolysis. Okay, so we did this then. Organic molecules and monomers, build them, dehydration synthesis, hydrolysis. We did that. Prokaryotic versus eukaryotic cell. Could you draw a simplified prokaryotic cell? Could you draw a simplified eukaryotic cell?
Yes.
Could you do that?
Yes.
What would a prokaryotic cell have?
Nucleus.
Would a prokaryotic cell have a nucleus?
No.
It have what?
It has like a nuclear
Nucleoid
Nucleoid region.
It's got like goo.
It's okay.
Like
It's
nuclear
okay.
goo.
It's okay. It's all right. It's all right.
It's okay, we're good. All right, so if I was going to draw a prokaryotic cell, it would have a cell membrane. It would have a whoa.
Looking.
It would have a cell wall.
It's gonna happen.
It's going to happen.
Oh.
You're magical.
And then it could have a what? Capsule around it. Okay? It wouldn't have any DNA in it, but it would have a what kind of region?
Nucleoid.
Nucleoid region. And what might these be? Ribosomes. Maybe it has a little bit of what?
Tail.
Flagella.
Flagella. But if I was drawing, it doesn't matter. I could draw a plant cell, or I could draw an animal cell. What does it look like I'm drawing right now?
Plant cell. Cells of the vacuole.
Vacuole. Then you have your chloroplast. Then you have your mitochondria. Wait, but that's the nucleus that you just draw with the nucleolus and the ER.
Rough ER. Good. And I would need maybe one of these.
Golgi body.
I do need one of those too.
And
What
you
do
need
you got?
chloroplast.
So that's the chloroplast.
Chloroplast. Yeah, I do need a Golgi body. Now, would I see a lysosome in a plant cell? No, because they're not really digesting. They're making the sugars. Right? But I would, if I was going to draw an animal cell, I wouldn't have this. I wonder if I can isolate this. Come on now. Come on now, baby. I wouldn't have this cell wall on here, right? I just have the blue cell membrane. I don't know if I can get rid of this. Delete.
No.
Boom. Okay, and what else wouldn't I have if this were an animal cell? I wouldn't have a chloroplast. Vacuole wouldn't be all huge, right? I can maybe make my nucleus a little bit bigger in proportion,
Yeah.
right?
You can have
I could have some
smooth ER and rough.
lysosomes, smooth and rough
Lys-
ER.
and you
Could
have-
you label what are really important structures?
Heck yeah.
Okay.
Delete those chloroplasts.
Okay, prokaryotic, eukaryotic cell, plant, animal, we did that. Structure of cell membrane and the functions of the components. Could you draw a cell membrane?
Yeah. You have the phospholipid bilayer with the hydrophilic heads and the hydrophobic
Okay.
tail.
Oops, wrong.
Oh. Yeah.
Those are the proteins.
Bro, I'm gonna drop the heck out of this place.
What's that I'm drawing right there?
That's for
Glycolipids, glycoproteins. What's this I'm drawing?
sure. Cholesterol.
Yeah, you couldn't even see it, but you knew. I'm so proud of you. Okay, and let's go over. We already did. You can't go through if you're large or charged. Let's go through all the protein functions. Ready? They are channels, carriers, cell recognition, receptors, enzymatic, and junctions. Junctions. Okay, so back here, let me see where we're at. Come back. Ways to cross a membrane. We did functions. Cholesterol helps with the fluidity.
Yes.
Phospholipids create a barrier.
Yes.
Proteins, we went over the functions. Ways to cross a membrane, we know.
Through the phospholipid bilayer.
Or used a channel.
What are the 3 kinds of endocytosis?
Oh, phagocytosis.
Stop. Don't race. What are the 3 types? Pinocytosis, which is cell drinking. Phagocytosis, which is cell eating. And then receptor-mediated endocytosis. And then the opposite of that is exocytosis. Good. Okay, let's talk about this. Active and passive. Active requires
ATP.
Yes, it does. ATP, energy. Do I do active transport across the phospholipids?
No.
No. What am I using ATP?
Proteins.
Protein pump or whole membrane. Endo and exocytosis is active transport. Passive transport is through the phospholipid. And when I talk about through the phospholipid, what things can easily move through the phospholipid?
Small and uncharged, non-polar.
Right, give me some.
Water.
Gases. Like what?
Oxygen.
Carbon dioxide. Good. All right, and then when you start talking about that, then you have to talk about osmosis, and then we could start talking about our cell membrane lab too, right? Osmosis, what we want to remember for that.
Jeez. Ooh.
Any day now.
I know, look at how slow it is.
I feel good.
Okay, so this is a terrible drawing, but what is that supposed to be right there?
Semi-permeable membrane.
What am I drawing right here?
Solution.
Some kind of solution. What would you describe this one?
Hypertonic.
And this is?
Hypotonic.
And we know water must flow from the hypo into the hypertonic environment, right? And if it were same on either side, what would you call that? Isotonic.
Because it's
And in our lab, the first part of the lab, we looked at a animal cell, your cheek animal cell, right? What it looked like underneath the microscope. The second part, what did you look at? An egg, right? And you put it in a hypertonic environment to get it set, and then a hypotonic, and you could describe for me what you did in that lab. Okay, and then the third part of the lab, beaker, and you put fluid in that beaker. What did you put in that beaker?
Iodine.
Iodine and
Distilled water.
Okay.
And
Then you had some dialysis.
dialysis tubing.
Okay. Then you had some dialysis tubing, and inside that dialysis tubing, initially, what did you put? Starch. Okay, good. And uno momento. Oh, I know what I want. Hang on, sorry. Spinning wheel of death. Okay, I'm there. Oh, I'm there. Why is it so slow? Okay, so out here, do you see that I'm touching that? Out here, what color was this out here? Amber. Okay, and initially, what color was inside the bag?
Clear white.
Clear white. Okay, and then over time, Okay, then over time, what color did it become in there? Dark blue. Why? Right, so there was evidence that iodine could move. It must be small enough to move because iodine went in the bag and hooked up with starch. Starch is too big to get out of that dialysis tubing because if it could get out, it would turn the whole beaker
Dark blue.
dark blue. And we know the sugar moved out because we tested it with
Our pH.
glucose test tape.
Oh, glucose.
Glucose test tape, yeah.
That's different than the pH test?
Yes, totally different thing.
Yeah.
pH measures the hydrogen concentration. We were measuring the glucose concentration. Okay, so we did cell membrane lab. We did it all.
Good
Is there
job,
anything?
We do like class lab.
Yes, we can.
Yes.
Let me see if I have one in here. Oh, look, these created images. Do you know what? Do you know?
Still question.
Oh, yeah, you can't see this question. Sorry. Here. What class are we in? Mammalia. Class Mammalia, right?
Oh, that's
Well, one do you think is right?
C.
C. Atom, molecule, organelle, cell, tissue. Yeah, what's wrong with A? Molecule, because you can see what's wrong with the other ones, right? So your answer there was C. Okay, good. What is it? Eukarya. Yeah, eukarya, A. All right. It's D. Yeah, D. Classification, you look at similarities between organisms and their evolutionary history. I did that. It's this one. The sun. The big yellow one's the
The
sun.
green plants, obviously.
Oh, you know what? Scientific method. I don't think we reviewed that. What do? We make
Observations. What?
Okay, good. Then what?
Question.
Ask a question.
Oh, collect data, search.
Do some research. Then we form a hypothesis. Do our experiment. Our experiment should be a what experiment?
Control.
Did you see how I was being controlled? And then what are we doing here? Analyzing our data and then make some conclusions. Yeah, claim evidence reason. Good.
Oh, the different types of hypotheses. I have a question.
Yes.
So, the two types of hypothesis is the if then because,
Research.
and then there's that second one which is
Null.
which is generalized.
Null is saying that there's not a connection between the independent variable and the dependent variable. It says there's no connection.
And you prove if there is.
If you get too much data to show the contrary, then you're supporting, you're not proving, but you're supporting the research hypothesis. Okay, what are all organs comprised or built out of? Tissue. Atom, molecule, organelle, cell, tissue, organ, right? Okay. Oh yeah, we didn't review this either. What is the mechanism for evolution called? We talked about adaptations. This is on when we went through the characteristics of life. This is part of that.
Natural selection.
Boom. Natural selection. That is the mechanism for evolution. Good job. What is the most inclusive category of the classification categories? What is it? I couldn't hear.
Domain.
Domain. Perfect. So the genus is just Homo. Species is sapien or Homo sapien. absorptive heterotrophs, fungi. Fungi, this is fungi.
So domain.
Kingdom.
Okay, I was right.
Yeah, remember, it goes domain, and there's 3 domains overall, right? And then within the domain eukarya, there are 4 kingdoms. So I'm asking you about that kingdom, right? There are 4 kingdoms. What are the 4 kingdoms? Animal, plant, fungi, and protista. This is usually unicellular. This is a heterotroph, but it's absorptive. A plant would be what?
Autotroph.
Autotroph. And this would be an ingestive heterotroph.
I remember this.
Remember that? Yeah. Could be autotrophs or heterotrophs. Protista. 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 You want another one? Death of a species or larger classification categories. Extinction. Good.
Yeah, it's true.
True or false? Now, where could you get more questions like this? Back of your chapters, right? What percentage of your test is going to be multiple choice? 80% is multiple choice. So you want to be able to practice doing multiple choice. If you were going to take your driver's test next week, what would you do?
Drive.
You would drive. Okay? So drive. Take multiple choice questions to practice for the multiple choice questions you're going to get tomorrow. Okay, how can you do well in the group part? You know I'm going to ask you about the lab in the group part. You know I'm going to ask you about an additional objective. You know I'm going to either ask you about a prokaryotic or a eukaryotic cell or a cell membrane, right? Big things we've drawn. You know I could ask you about any of the 4 important organic molecules. Those are big things we've studied. So be ready for that on the group part. Okay? Questions, concerns?
Qualms.
Okay, remember, do the hardest part first. So go home and get at least some chunk, whatever's your weakest of your chapters, spend a little time looking over that. You've made some beautiful notes. Look over your notes and then go take some practice questions. You could read the summaries at the back, right? But make sure you do some practice multiple choice questions. You're super smart, and if you get tired, have a piece of toast. Okay, make good choices.